Hi everybody, it's Dr. Burgess here, and this is a run through of your combined science biology mock exam. So this is for higher tier, and hopefully it will give you a good idea of how you could improve. Now I realise you don't have your exam papers with you, but you'll have your analysis sheet, and you probably can remember the questions. But I'll go through all the questions and all the answers and explain some of the answers too. Okay, so first question is related to the topic cell biology, and this is osmosis, and this is the required practical for osmosis. So let's look at the first answers. Okay, so it says it gives you the method, so it investigates the effects of different concentrations of sugar solutions, and then it gives you the method, and it says what is the independent variable. So the independent variable is the thing that is changed, and you can see in that they say they are investigating the effects of different concentrations of sugar. Okay, so that is the independent variable. Okay, so that's the thing that's being changed, so it's the concentration of sugar solution. Then it goes on to 1.2. It shows you some data, so it shows the results. You can see the mass of the potato at the start, after, and then the change in mass. And it says explain why the potato in 0.0 molar per dm cubed sugar solution increased in mass. So you can see here, and you can see that it increased by 2.2. Okay, and it's 2 marks. So you'll notice that it says what happened. Well, you can see that it gained in mass, but also you've got to explain. So you've got to give a reason why. Okay, and so basically it's due to osmosis. So it gained water by osmosis, or you could say the concentration of water outside the potato is greater than inside the potato itself. But so you're describing that it gained water, but you're explaining why, and that's because of osmosis. Question 1.3, okay, they give you a graph, okay, and they say some of the results have been plotted for you. You should plot the data from table 1, draw a line of best fit through the points. So let's have a look at how you would do that. So here's the data, so you can see the data up here, and we've got to plot the 0, 0.1 and 0.2. So the first one is 2.2, so you can see that it is around about, well, not around about, it is right there. I'm using a graphics tablet, so just getting the hang of it. 1.15 at 0.01, so 1.15, so it's just in the middle there. Okay, just there. And 0.25 for 0.2, so 0.25, so 1, 2, just in the middle there. Okay, so I've plotted the data, not very well. You probably could plot the data better with a pencil. And then it says draw a line of best fit through all the points. Now the line of best fit is actually curved in this case. So I'm going to start from that point there, and I'm going to try and go through as many of the points as I can, and you can see that it curves like that. Okay, so some of you are drawing point to point. Don't do that. It's a curve of best fit. Okay, and that's how you would do that question. It then says the mass of the potato does not change when the concentration of solution inside the cells equals the concentration of solution outside the cells. So there is no mass, there's no change. So determine the concentration of sugar solution. So we're basically looking at this point here, 0. Okay, and what you need to do is get from your line of best fit wherever it bisects that line, and in this case it's 0.2678, 0.28. So whatever your line showed, that is the result that you would give. Okay, so on to 1.5, and you'll notice that they repeat the table because you're going to use the table. It says calculate the percentage change in mass for the potato in 0.2 sugar solution. So we're looking at that one. Okay, and it tells you here's the equation. All right, and it gives you the equation that says give your answer to 3 significant figures. So the change in mass, well, luckily they've already done that for you, so you have to work it out. So it's 0.25, and then divided by the mashed potato at the start, which is 7.96 times 100. So that gives you a long number, okay, but it's three significant figures, so it's 3.14, and that is the answer for three significant figures. So starch is digested to form sugar molecules, so this is about enzymes and different enzymes in organisation. So the enzyme that digests starch was amylase. Where are the most food molecules absorbed? You should know that. Some of you didn't. It is the small intestine. And then 1 mark, it says give one advantage of using an electron microscope compared with using a light microscope. So you did this in cell biology, and there are 2 answers you could have. So let's have a look at the answers. So you could have had anyone from a greater magnification or higher magnification or a higher resolving power. Okay, so again, that's your knowledge. So if you revise that, you would have got that correct. Okay, 2.4, what type of blood vessel is labelled X? So let's look at X. Well, that looks like, to me, looks like a capillary. So it is a capillary, so you would put the correct answer, capillary. You tick 1 box. If you tick 2 boxes, you won't get it right. Okay, so then another calculation. So this is related to cell magnification. Tells you the real length of one villus is 0.8 millimeters. Calculate the image length if the villus is viewed at a magnification of times 20. And it actually, again, it gives you the equation, so that's really, really helpful. So let's have a look. So what I've drawn is the IM triangle. So we want to calculate the image length of the villus. Okay, so it gives us magnification. You could use that, but I remember teaching you the IM triangle. So we need to find out the image. So it's actual times magnification. So in this case, it's 0.8 times 20. Actually 16, and it's in millimeters, so you don't have to convert it or anything, and that is the answer. Nice and simple. Some of you overcomplicated that question, so just need to remember the IM triangle, and then there you go. So this next question shows 2 cells from the surface of a villus, sugar molecules inside and next to each cell. So it asks you to name the process. So this is transport across molecules in cell biology. So A, so how is it getting from A, so outside, inside? Well, that would be diffusion, because it's going from a higher to a lower concentration. So it's diffusion. And name the process by which sugar moves into cell B. So you can see there's a low concentration there, and there's a higher concentration, so it must be active transport. So it moves from a low to a high, and then give one use of sugar in the body. And the main one is respiration, so using glucose is respiration. There we go. So you could also have to form glycogen or make amino acids and proteins. They would also allow as an energy source, but it cannot be accepted if you said to create or produce energy. Okay, it's not producing energy. All right, it's not creating it either. So it's releasing energy, or it's an energy source. So that's what you would need to include in your answer. Right, 2.9, so there's 4 marks here. They show you a villi are adapted for efficient absorption of sugar molecules. Now, you talked about villi when we did digestion, okay, but also this is related to gas exchange, and you did that in cell biology as well. So you don't need to know too much about the villi because they give you a labelled diagram, but it does say, let's just use my highlighter. It says explain how the villi are adapted for efficient absorption of sugar molecules. You get 4 marks. So this is about gas exchange. So you've got to mention the structure, and then you've got to explain why that structure is useful for the efficient absorption of sugar molecules. So before I go through the answer, I just want to show you how this would be marked. So it's split into level 1 answers and level 2 answers. Okay, and particularly if you notice down the bottom here, it says for level 2 you must make links between the structure and its function. So you can't just mention the structures, you've got to explain and say how does that help absorption of sugars. Okay, so the content doesn't need to include all of these things, but it needs, for 4 marks, as it says here, you need to identify relevant points. They need to be given in detail and logically linked to form a clear account. Okay, so you would have to write in sentences. One thing that I've noticed some students are doing is writing the question. So when they start their answer, they're saying the villi are adapted for efficient absorption of sugar molecules because, and that's not going to help you. You need to get straight into the answer. It's just going to waste a line of your exam and waste your time as well, because it's not going to get you any marks. It's important to think what you're writing is about getting marks. So if you look at the structures, so they show you actually in the diagram, if you knew nothing about villi, you can see here on the villus, on one villi or villus, they then have cells, and those cells have lots and lots of microvilli, and you can mention that they have many microvilli. Why is that useful? Well, it increases the surface area and speeds up diffusion. The walls of the villus, they're only 1 cell thick, or they're very thin, so that means the capillaries are close to the surface, so you've got a short pathway through which the molecules can travel. You've got a good blood supply, so the blood supply is maintaining a diffusion gradient. And also cells, if you notice, it does show you there, cells have mitochondria. So that's a clue that active transport is occurring. So respiration takes place, energy is being released or transferred, active transport is occurring. Okay, so there's lots of clues in that diagram to allow you to answer that question fully. Right, question 3. This was related to health and non-communicable diseases. Okay, and it says being overweight can affect the health and life expectancy of a person. Give 1 disease related to being overweight, and there will be lots of diseases. So it already mentions in the actual question about obesity, so you can't have obesity because it's mentioned in the stem of the question there. Okay, any other diseases though? Diabetes, you can't say type 1 diabetes. If you just say diabetes, that's fine. If you say type 2, that's better. Any form of heart disease, cardiovascular disease, you can't say heart attacks or heart failure. You've got to talk about disease, all right? Then high blood pressure, cancer, depression, and any other form of disease related to being overweight. Okay, then they talk about body mass index. We mentioned body mass index, but we don't tell you how to calculate it, and therefore they've got a equation. So they tell you woman has a BMI of 27 and a body mass of 68.1. Calculate the woman's height in meters. So you've got to fill in the information you've got. To find the height, it would be the body mass divided by the BMI. Okay, and that would give you 2.522 recurring, or 2.52. Okay, and then what you've got to do is square root it, because it's height squared, and we want to know the height in meters. So you square root 2.522. Okay, and then it's rounded to 1.59. But it says here, if 2.5 is given in step 2, allow an answer of 1.58. If it's not, then it's got to be 1.59 for that mark. So bit of maths there. Remember, there's about 10% of maths in biology GCSE in your paper. Right, 3.3. Okay, they give you some information on a graph. Shows a height body mass chart for children for adults, sorry. And it says, which weight categories describe the woman in question 3.2? So question 3.2, so height is 1.59. The body, the weight, body mass, sorry, is 68.1. So we need to look at 68.1, and the height we know is 1.59. So here we go. So approximately there, 1.59. So they are overweight. Okay, people are encouraged to control their body mass with diet and exercise. Describe, so it's saying describe, which is say what you see, okay, about how the balance between the mass of food eaten and the amount of exercise a person does controls body mass. It's for 3 marks. I think many students did struggle with this question, so I'm going to try and explain it to you. So the best way to do is kind of split up the question. So it's about the mass of food eaten and the amount of exercise. So if you eat more food related to energy, okay, so we're talking about the amount of the balance between mass of food and the amount of exercise. So if you increase your energy intake, if more food is eaten, okay, so also you're going to decrease your energy intake if less food is eaten. If you exercise more energy is transferred or released. You could mention that there's more respiration or higher metabolism. If more energy is taken in than is transferred, the body mass then will increase, okay, or you could say conversely, if there's less energy is taken in, then body mass decreases, okay. If energy intakes is the same as the energy transferred, your body mass will stay the same. So what you've got to try and do is split up that type of question. So think about if you overeat, if you undereat, if you stay the same, what will happen to energy intake and therefore your body mass, okay. I think some of you did struggle with that question, so it might be worth looking at that a bit more carefully. Right, on to question 3.5. This is a 6-mark question. Okay, so let's have a look at this 6-mark question. So it says, during long periods of vigorous exercise, the body respires anaerobically. Explain, so give reasons why, the changes that happen in the body during and after exercise. And that's the key thing there that some of you again missed. You've got to talk about what happens during exercise and after vigorous exercise. So before I talk about the answer, notice how it's marked by the examiner. So you get a level 1, okay, points are stated simply but their relevance isn't clear. A level 2 answer for marks 3 to 4, so they identify their attempts. The resulting account is not fully clear, so you've got to write in sentences. Level 3 though, the relevant points are identified, given in detail and logically linked to form a clear account. I'm also going to point out the bottom of the mark scheme before I talk about the actual mark scheme. So it says for level 3 you need to reference to changes during and after exercise, but also you need to talk about lactic acid and its removal. So the kind of content it's going to need, it's not going to need all of this content, obviously you're not going to be able to write all of this, but it needs a good relevant points need to be identified. So during exercise, some of the things are increased breathing rate, increased breathing volume. Why? Well, you're taking more oxygen for respiration, you're exhaling more carbon dioxide. You could mention that heart rate increases, so that increases blood flow to the muscles. It's transporting oxygen and glucose faster, so you're getting increased rates of carbon dioxide removal as well, and glycogen converted to glucose. So that's what's happening during exercise. Then you could talk about, well, what happens, because they do mention the body respires anaerobically. Well, insufficient oxygen means that lactic acid is formed, an oxygen debt is created, muscles become fatigued or tired, and then you could mention sweating or increased body temperature. So that's during exercise. Obviously, again, you can't mention all of those. I would mention lactic acid though, particularly for level 3, because after exercise, obviously your heart rate will remain high for a while, or it might slowly decrease. You're going to breathe rapidly. Some of you mentioned that you need to pay back an oxygen debt. So that means that you're breathing and getting your oxygen levels back to normal. And the reason for that is to break down lactic acid. Okay, so then you could say lactic acid is transported to the liver and it's converted back into glucose. Okay, so that's about the effects of exercise and respiration, anaerobic respiration. So there's quite a lot in there. But to get 6 marks, you've got to mention both changes during and after exercise and lactic acid and its removal. Right, question 4, cells. So this is cell biology, and it says describe 4 differences between a bacterial and a plant cell. So this question very much was about your revision of prokaryotic and eukaryotic cells, particularly difference between bacterial and plant cells. So you could either talk about plant cells, but in this case it's talking about bacterial. So first of all, they're smaller, they don't have chloroplasts. Bacterial cells don't have a nucleus inside a nucleus. They may have plasmids, which plant cells don't. They do not have mitochondria. The cell wall, both have a cell wall, but the bacterial cell wall is different because the plant cell wall, you'll know, is made of cellulose. Bacterial cells does not have a large permanent vacuole. And also, the bacterial cell does have ribosomes, but they are smaller. Right, so that was a bit of cell biology, and then it went on to infection and response and talked about gonorrhea. So gonorrhea is a bacterial disease. A new vaccine is being developed. Describe how a vaccine would work to prevent gonorrhea. So this question is very much about, well, how do you make a vaccination? So how does the vaccine work to prevent gonorrhea? So again, most of you knew what a vaccine was made of, so dead, inactive or weakened forms of a pathogen. Okay, and then that stimulates white blood cells to produce antibodies. Some of you were talking about antitoxins and things like that, but that's not correct. It's they produce antibodies. Okay, and then they circulate around your blood. You could mention that they remain in your body as memory cells, and then when you are exposed to the disease, so gonorrhea, okay, correct antibodies are made very quickly. Okay, and then the bacteria or the pathogens are killed and do not produce large enough populations to cause the disease. So any 4 of those. So the second half of question 4 then talks about salmonella and food poisoning, and it tells you chickens are vaccinated against salmonella to reduce the number of cases of food poisoning in humans. So it says explain how vaccinating chickens reduces the number of cases of salmonella poisoning. So let's have a look. Okay, so how, it's not asking how does it vaccinate, it's asking, well, what does it do? Well, it means then that there are fewer bacteria in the chickens, the eggs or the food, so therefore less is ingested by humans. Or you could mention there's fewer bacteria, so fewer toxins are produced. Okay, so you're not talking about immunity, you're not talking about how the vaccine's made, you're talking about, well, what's the result? And it's fewer bacteria in the chickens or eggs or food, so less is ingested. It's as simple as that. Some of you got very kind of confused with what it was asking. 4.4, give one way, so just one way that the spread of salmonella food poisoning from one human to another is controlled. And I think, again, one of the key things there is it's the spread from one human to another, okay, is controlled. So not eating the chicken, it's not about eating the chicken, it's about from human to human contact. It does say do not refer to vaccination in your answer. So don't mention vaccinations. Okay, and the main thing is that you should wash your hands before preparing food. okay, or wash hands after using the toilet. If you just said wash hands, that's what we call an unqualified answer, and it wouldn't get the answer, wouldn't get the mark, sorry. Allow good food hygiene, allow clean areas where the person has been ill, don't shake hands with someone who has food poisoning. So those kinds of things related to human, from one human to another. Right, 4.5, the number of cases of salmonella food poisoning is usually higher in summer than in winter. Suggest one reason why. So this question was about summer, not winter. So you're not talking about bacteria in cold conditions, you're talking about bacteria in warmer conditions. So in this case, warmer weather, so the bacteria reproduce or increase faster. Some of you mentioned about barbecues, that was fine, saying the food is not cooked properly. There are more barbecues in the summer than the winter. On to question 5. So this was related to photosynthesis. This is bioenergetics and food production as well. So the first one says, how can oxygen production be used to show the rate of photosynthesis? So this was very much related to the required practical. And basically, for 1 mark, you're measuring the volume of oxygen produced in a given time, or when oxygen is produced in a given time, the rate of photosynthesis is faster. So you talk about the rate. So if there's more oxygen, there's a faster rate of photosynthesis. Okay, you need to mention gas, or in this case oxygen. So you need a reference to rate is needed for this question. Right, they then give you a graph. This is related to limiting factors, so you can see different levels of CO2 at different temperatures, and it tells you the percentage of carbon dioxide in the Earth's atmosphere is 0.04. So that's, you can see that figure is, you haven't added any CO2. So it says name the factor that is limiting the rate of photosynthesis at point X. So you can see at X, this is related to light intensity. Okay, so if you increase the light intensity, the rate of oxygen production increases. If you decrease it, then if you decrease the light intensity, it decreases the rate of oxygen. So the name of the factor that is limiting the rate, well, it must be light intensity. So you need to mention light intensity. If you mention light, that's fine. And that is the answer for that question. Question 5.3 then goes on to talk about farmers growing tomatoes commercially, okay, to try and control the rate of photosynthesis and make maximum profit. And it tells you a farmer can control the temperature, carbon dioxide concentration in a greenhouse. So that information is really important for the rest of this question. So then it says, what is the minimum light intensity a farmer should use to get the maximum rate of photosynthesis, shown in figure 5. So let's have a look at figure 5. So the minimum light intensity to get the maximum. Well, we know the maximum rate of photosynthesis is when you're getting a maximum rate of oxygen. So basically at this point here, so light is limiting all the way down here until we reach plateau. Okay, then some other factor is limiting. So if you look at the graph, it's still increasing all the way up to that point there. Okay, so if you read it down, you'll notice that it is 20,000 lux, and that would be the answer. So it's 20,000. There we go. Right, then it says the light intensity you gave in question 5.3 may not give the farmer maximum profit. Explain why. Be honest with you, I didn't like this question, to be honest, because it reminds me of business studies, but there you go. So may not give the farmer maximum profit. Okay, well, it might not give them because there is a cost. So you're going to be spending more on heating the greenhouse. Okay, so if you notice, it's 30 degrees at that point there, Okay, there is a cost for increasing the carbon dioxide in the atmosphere. Okay, there is a cost as well for lighting in the winter. So therefore, it's going to affect the amount of profit. Okay, or costs should not be recovered, could not be recovered by increasing the sale price of tomatoes. Right, this next question was quite difficult. So it does say, explain the results when the light intensity was 0 lux. Well, if you notice 0 lux, okay, it's actually at -2. All right, so it's not producing oxygen. So the oxygen is being used by the plant. It's not 0, it's -2. So it means that the oxygen is being used by the plant. So what's it being used for? Well, it's not being used for photosynthesis because there's no light, but it is being used for respiration. So I think the first 2 points most people got, where there's no light, there's no photosynthesis, so no oxygen is produced. However, it's being used. So what's using it? Well, respiration is using it. Okay, so you need to mention that respiration is using it, and you do need to mention that oxygen is negatives. Okay, so you can't just say it is -2. You have to say overall oxygen production is negative. Quite a difficult question there to get full marks. On to question 6 now, and this is related to infection and response, and this is related to drug trials. So they give you some data here, and they say how much more time, there you go, more time did the clinical trials take compared to preclinical testing, and they give you a selection. So we need to first of all work out preclinical. So if I draw a line down here, not a very good line, that is 3.5 years. And for clinical trials, so it's phase 1, 2 and 3, it tells you which is clinical trials, which is 10. So 10 minus 3.5 is 6.5. Okay, now that's not the answer because it's how much more time. So 6.5 minus 3.5, and that gives us 3. So it is in fact 3 years. So question 6.2, so looking at phase 1 clinical trials, low doses of the drug are tested on healthy volunteers. And it says suggest one reason, one being in bold, why low doses are used in phase 1 drug trials. And basically there are 2 answers you could have, either to reduce any risk or to look for side effects. It would also accept if you said there was less harm will be caused with a lower dose. Okay, you can't just say it's unsafe, all right, or dangerous, unqualified. So that means you've got to give a reason, and that's to reduce any risk. It's not related to side effects because at this stage you're testing with healthy volunteers. So it's not about unknown side effects, I should say. So it's look for side effects. Then suggest 2 reasons why healthy volunteers are used in phase 1 clinical trials. And I think it was quite difficult to get both marks for this. You might have got 1 mark for it because it's too great a risk for an ill person. Okay, so you could argue if you used people who are already ill, they might get worse. And also if they basically the patient might be taking another drug, but also you could mention side effects as well. But side effects of drug are easier to identify. You can't just say side effects. 6.4, the result of clinical trials can only be published after peer review by other scientists. Suggest 1 reason why the results must be reviewed by other scientists. And really they're looking for key things like it's to prevent false claims, it's to make sure the results are not biased, okay, to avoid bias, to check that the conclusions are correct or valid. You couldn't mention anything related to accuracy, reliability or precision, because it's not talking about actually performing the clinical trial, it's after the clinical trial. So it's one of those three basically. And then 6.5, they have lots of information about treating drugs with a clinical trial, and they talk about the different types of drugs that they can use to treat AMD. So they give you lots of information in there, and that information they want you to use in the question. So the question says, in 2018, the High Court in the UK gave permission for drug C to be used to treat AMD. Evaluate, and I will highlight that now, evaluate the decision to allow the use of drug C to treat AMD. So remember what evaluate means. So evaluate is looking at basically the pros, so the advantages and the disadvantages. Okay, so what are the advantages and disadvantages? And you need to mention both in your answer. There we go. So let's have a look at the answer. So it's a 6-mark question. It's going to be split into levels. Okay, so 1 to 2 marks. Level 1, relevant points are made, they're not logically linked. Level 2, some logically linked reasons are given. There may also be simple judgments. And if you look at the bottom here, it says need advantages and disadvantages for level 2. So if you only gave advantages, you're limited to 2 marks. Okay, so let's talk about the arguments for use. Now, again, you can use some of the information they've given you in the stem of the question, but you can't just copy down that information. You've got to do something with that information. So things like, the first thing is, I think most of you don't realise that drugs are bought, if it's for medical use, most drugs, they're not bought privately by people, they're bought by the NHS. So the NHS buys the drugs and then they're prescribed through your GP or hospital. So it will save the NHS money for drug C. And then here's some data. So you could say that 20 times as many people or 19 more people can be treated compared to drug A. Now some of you gave figures, so you actually worked out how many extra people could be given the drug, which is good. Also 29 times as many people can be treated compared to drug B, so that's comparing C with A and B. So you could also say more people are treated for the same cost, patients are treated sooner, improves patient choice, used in other countries, so it's likely to be effective, and also it's likely to be safe. Arguments against use. Well, the main thing is it's not tested in the UK. You might not be able to check that it's as effective as drug A and drug B, so you might have to take more of it, so you're not saving cost in the end. Is it safe to use? Are there unknown side effects? Could the doctors be confident in prescribing drug C? It could go against the laws regulating drug development in the country. Also, has it been fully tested? It's also not been licensed or approved in this country. So there's many different reasons for and against. You don't have to mention all of them, but a judgment is made by the examiner on whether you've made strongly linked and logically supported kind of judgments using a sufficient range of correct reasons. So I would say probably we're looking for 3 advantages and 3 disadvantages, all written up really nicely and logically and correctly. Okay, so that brings us to the end of this analysis. I hope you found it useful, and I'll see you soon.